Thanks for that introduction, Robin, and hopefully you can hear me all right with this microphone. Okay, good. So yes, I put this slide up here just to emphasize that we are changing a bit from what we're talking about, quite a bit. Moving out of the oceans up onto the land now, and I'll be giving this lecture today, just an introduction to the terrestrial environment, and I'll also be focusing on two cycles which are of particular interest and related to this talk we just had from Mathieu. The nitrogen and phosphorus cycles, which have a lot to do with nutrient loading. So I'll try to give a bit of the biology behind why we do have nutrient loading into, say, river systems and how human agriculture is really affecting that process a lot. So that's what I'll do today. And then on Friday, I'll give another lecture talking a bit more about what Robin said I do in my lab, which is working to create a new wave of cereal crops that are nutrient-enriched and have vitamins and minerals that humans need as part of their diet. So let's just get started. So yes, we're shifting our focus now up to this 25%, or about 30% of the world is land. Most of that is covered by plants, as you can see from this photograph. About 25% of the Earth's surface is covered by plants. They're one of the dominant drivers of biogeochemical processes, and so that's what we'll be focusing on. And when we talk about biogeochemical cycling in the terrestrial environment, we very often talk about plants because they play such a major role. We also talk a lot about soil microbes because they are doing interesting things in the soil which affect, they're doing biogeochemical processes, but they also interact very strongly with plants to participate in the uptake of nutrients. So they do symbioses. And a good example is this right here. These are roots of clover. And you see all those little nodules all along that root. There's bacteria inside of there, and they're fixing nitrogen from the atmosphere and giving that to the plant. And so we have these types of symbioses that occur for the uptake of a lot of different elements for nitrogen and phosphorus. So that is why we often talk about plants and microbes when we talk about biogeochemical cycling in the terrestrial environment. And like I said just a minute ago, the terrestrial and the aquatic biogeochemical cycles are very closely linked because a lot of what we do on land, particularly in agriculture, 40% of the Earth's land is now used for agriculture. What we do in agriculture very much affects biogeochemical cycles in water, in rivers and in oceans. And when I talk about the nitrogen and phosphorus cycles, you'll see that some of those impacts of agriculture. So the outline that I have for today, hopefully I can get through this by the time we have coffee, is just four things I wanted to talk about to introduce this terrestrial system with this focus on plants. First of all, I wanted to talk about just nutrients that are essential for terrestrial plant life, and I'm not going to spend a lot of time on this, don't worry. But I think it is important to go over what these nutrients are because these are the ones that plants are mining for in soils, and therefore they're very important for biogeochemical processes. You probably had this information in first-year biology, so I'll go pretty fast. Then the second part, again, I'll go fairly quickly, which will talk about nutrient uptake by plants. How do plants actually source these nutrients from the soil? Just really quickly go through that. And some basic factors that affect the availability of nutrients in soils. So how do various factors related to soil chemistry affect plant uptake of nutrient uptake by plants. And then I'll get to these two things which I talked about before, the terrestrial nitrogen cycle, and introduce that, then talk about these anthropogenic changes as a result of agriculture, which have really altered the nitrogen cycle on land. And then the same for phosphorus. So those are the four areas. So let's first talk about these nutrients. What are the nutrients which would be important for plants and therefore important for biogeochemical cycling? There we're talking about essential nutrient elements. And so plant biologists have worked this out over the last 100 years or so, usually with hydroponic experiments where they just culture plants in hydroponic conditions and just add and take away elements and see which ones are essential. And so it's very basic. What is an essential nutrient element for any organism? It's in its absence, a plant cannot complete the normal life cycle, and or the element is part of an essential plant constituent or metabolite. All right, so that's what essential nutrients are. When we look at plants, we generally agree that there's about 19 elements make up a plant. So these are the elements which are crucial for metabolism and for all the life processes of plants. And so I've listed them right here, these 19 different elements. I'm not going to talk about these elements at the top here, hydrogen, carbon, and oxygen. You can see plants need lots of those. All living organisms do. But those will be covered tomorrow when Peter Rayner is talking about carbon cycling in the terrestrial environment. So I won't talk about that. But what I will cover is these groups here. So we have these other two groups of elements which all come from the soil, and we call these our macronutrients. And our micronutrients, and as the name implies, that just reflects how much is within the plant tissue itself. And if we look at these macronutrients, you see the first one listed there is nitrogen. Nitrogen is required in really large quantities by plants, and it's usually the primary limiting element in the terrestrial system. And in fact, if you just look at hydrogen, carbon, oxygen, and nitrogen alone, that's about 97, 98% of what makes up a plant. So just those four elements alone contribute a lot to what a plant is. But there's also some very other important macro elements listed there. For instance, we have potassium. Potassium is particularly important for plants because it's what enables them to take up water. So they regulate the flow of water into their cell tissues. Likewise, the flow of water out of their tissues, usually with potassium. So we call that osmoregulation. And plants, again, need a lot of potassium. Then we have calcium, magnesium, phosphorus. Phosphorus is critical for the plants, and I'll discuss why later on. Sulfur and silicon. So those are our macronutrients. And when you look at any type of fertilizer, I'm sure all of you know this, but you have three numbers which are always listed on the bag of a fertilizer, and that refers to NPK, nitrogen, phosphorus, and potassium, because we know that these are extremely limiting nutrients for terrestrial plants, and so you need to add them in very large quantities. And it's the large quantities of nitrogen and phosphorus that we've been adding to plants now for nearly 100 years, which are really changing biogeochemical cycles in the terrestrial environment. Then below that, we have our micronutrients, and you can see a whole bunch of them there. I won't read them out, but one that I'll talk about on Friday when I give that lecture is iron. Iron is the micronutrient which is typically required in the highest concentrations by terrestrial plants because it's very difficult for plants to take up. Even though iron is the fourth most abundant element in soils, it's very insoluble at biological pH. And so basically plants all around on land surfaces are usually fighting to take up iron, and they've evolved some very interesting mechanisms to take up iron. So I'll mention that on Friday. But those are the two broad categories, macronutrients and micronutrients. This is how nutrients were initially classified by plant biologists and plant physiologists. Nowadays, people think that's a bit too simple. It's better to actually classify nutrients more according to their biochemical function, and that's why I have a few slides here just to tell you what those groups are. We generally say there's group 1 now. These are nutrients that are part of carbon compounds. Makes sense to classify nutrients that way. And we just have 2 there. We have nitrogen and sulfur. And these are really important elements, of course. Nitrogen goes into amino acids. That's why all organisms need so much nitrogen, so that we can build proteins and nucleotides and all types of things. Group 2 would be nutrients that are important for energy storage or for structural integrity. And phosphorus falls into that category. And as we all know, plants don't need quite as much phosphorus within their tissues, but they have to have it, absolutely, to build things like cell membranes. So here's a picture you all remember from first-year biology, I'm sure, of the phospholipid bilayer, which is what all biological membranes are composed of, and you can't build that without phosphorus. So that's what group 2 is. There's phosphorus, silicon, and boron there. Moving on to other groups. Group 3 would be nutrients that remain in ionic form. And there's quite a few nutrients in plants that remain in ionic form. The first one up there is one of those main ones from fertilizer. It's potassium. And like I said, potassium is really important for osmoregulation, so flow of water in and out of plants, keeping them hydrated. Also, for them to open and close their stomata. That's all done by just moving potassium in and out of their stoma. Also, a lot of these are important as cofactors for enzymes. And so that's why I put this little picture here just showing that a lot of times enzymes can't function without some of these elements that remain in ionic form, like potassium or chloride. And then you have some other ones here, such as magnesium, which also remains in ionic form, and you can't build chlorophyll without this magnesium, because right at the center of the chlorophyll ring, you have magnesium. Okay, so that's group 3, nutrients that remain in ionic form. And then finally, in group 4, you have nutrients that are involved in redox reactions. And that makes sense as well to separate these guys out and keep them in their own group, because redox reactions drive so many of life processes. In plants, they drive photosynthesis. And so you have iron right up there at the top, because it's very involved in these redox reactions like photosynthesis, which I've put in a very simple form right there. And like I said before, iron is usually the most limiting micronutrient in plants. And then you have these other ones like zinc, copper, nickel, and molybdenum. All right, so that's nutrients in a nutshell that are important to plants. I told you I'd go through that pretty quick. Now we'll go through, just briefly, how do these nutrients get into the plant in a terrestrial environment? They need to have mechanisms to take these up, and likewise to keep things out that they don't want to accumulate in their tissues. There's a lot of harmful things in soils, like cadmium, that plants would try their best to keep out. So first of all, when we talk about the plants growing in their terrestrial environment, it's through the root system, obviously, that all of these 16 different elements are going to come through. So here's a nice little picture of a plant, and it's in this root system where all of these minerals are going to get into the plant. And here's another classic picture of just a cross-section, transverse section of a root, which you probably remember from first year biology, by making these sections yourself back in those practicals in first year. And basically what we say is that mineral ions cross the root apoplast and the symplast. What does that mean? Apoplast is the non-living parts of the root. So you can have a little physical crack in the root, and minerals can actually just start to flow in there and water. Or the symplast, and the symplast would be nutrients being taken up by a cell. For instance, here's a root hair. Minerals can be absorbed across the plasma membrane and go straight into that cell and then work their way through cells. Okay, so they can come in initially through the apoplast, the non-living way, or the symplast, the living way. However, everything has to enter the symplast at what we call the Casparian band. Is that a word that people remember? I'm not sure what your backgrounds are, actually, so I'll just explain that Casparian band is right here. It's what defines the central part of a plant root, and it's a living layer. It's very waxy. Things cannot just diffuse across. Everything needs to be transported across that layer. And so this is the way that plants can hyperaccumulate things they want to take up, so nitrogen and phosphorus or potassium. And likewise, they can decide to keep things out at this Casparian band by not transporting it across. And so this is just a blown up diagram of that Casparian band. And that is the symplast. And I'm not going to go into this in much detail at all, but on that Casparian band, all across those cells, you have transporters that are specific to these 19 different elements. Okay, so you'd have nitrate transporters, you could have sulfur transporters. Everything's moving. A lot of things are moving down their electrochemical gradient, and so all you need to do is just open up a channel protein, or you can have carrier proteins, and they just take them, escort them across that phospholipid bilayer down their electrochemical gradient. However, sometimes organisms need to accumulate something against its electrochemical gradient, something that's very sparse in the environment. And so we do primary active transport, which, again, things we talk about in first year biology. So that's moving something against its electrochemical gradient. And what do we typically move across? We usually move across hydrogen ions or calcium. Those are usually the two things we do. And then, once you've moved those hydrogen ions or calcium against their electrochemical gradient, you can let them back through, and they can carry something else, okay? And that's what we call secondary active transport. And secondary active transport is what we use to accumulate a lot of ions. And so here again, that's an example of that. You can just see hydrogen ions have been pumped against their electrochemical gradient. Then here we're letting them back in. And as we do that, we couple it by moving this solute against its electrochemical gradient. And that is how we accumulate quite a few different nutrient elements in plants against their electrochemical gradient. Secondary active transport. So that was all I was going to say about how these elements get into plants, just because I think it's worth mentioning the terrestrial environment. Plants have to have mechanisms to pump them into the tissues. Just a few very basic things now about the chemistry of soils that affect nutrient availability. The most basic one would be parent material, right? It's so obvious, it's questionable whether I should even mention it, but obviously the rocks or the sediments that give rise to the soil determine many aspects of soil fertility. And plants, it's different than the aquatic environment. They don't just have water circulating around them, they're stuck wherever they've germinated and where they're growing. And so a lot of the nutrient characteristics are going to be determined by the soil that they've germinated in. For instance, I've just given some examples here. If it's soil derived from granite, it's usually low in P and other cations compared to limestone. Large parts of the world, particularly in the subtropics, are serpentine soils. And when they give rise to soils, those soils are really high in heavy metals and low in things like phosphorus. So just the parent material has a big effect. There's a couple generalizations we can make about parent material. We say in relatively young landscapes that follow recent volcanic activity or glaciation, phosphorus availability is relatively high, and nitrogen tends to be the limiting element there. However, in very old soils, like the soils here in Australia, phosphorus is often the limiting nutrient there. And that's the case here in Australia. I mentioned that a little bit later in this lecture. Soil pH is also something that has a massive effect on the availability of nutrients, which I'm sure many of you know. Plants try to keep their pH at about 5.5 to 6.5. That's usually the optimum range for them to absorb nutrients. If you move down into the acidic range, plants can be flooded with a lot of metals, for instance. The oxidation state of metals is very low at a low pH. So you'll get, say, a ferrous, iron, Fe2+, and those things can flood into a plant. So at very low pHs, around 4 or 3, you can get toxicity of minerals. Likewise, at high pHs, it's very difficult for plants to absorb most elements. And metals, it's almost impossible for plants to absorb metals once the pH gets above about 8. So whether the soil is acidic or alkaline has a big effect. So here we can see you can get molybdenum deficiency at a low pH. Likewise, you can get an iron deficiency at a high pH.
So what determines the pH though? Is that the parent rock?
Yeah, a lot of that is determined, yes, by the parent rock or also fertilizer applications. If you've been doing that for 100 years, you can lower the pH or raise it, depending on what you're doing. But it's, I believe, largely just determined by parent material. Yep. And there's interesting things that plants do. They do, to a large extent, they have to sit there and they have to tolerate what parent material they've been given. But plants can modify their rhizosphere quite a bit. So they can, in some cases, change the rhizosphere pH, which is pretty interesting. And why would they do that? Well, the solubility of iron, like I've said, is very affected by pH. It decreases 1,000-fold for each unit increase in soil pH. Magnesium, copper, and zinc, they also decrease 100-fold for every increase in pH. So if a plant wants to absorb those micronutrients, it will try to lower the pH. And plants can strongly reduce the rhizosphere pH by excreting things, and they typically excrete organic acids or protons. And there's been lots of experiments. I'm not going to show them here. But for instance, you can show that sunflower, if you put them in a pH of 7, that's not a very good range for them to absorb a lot of micronutrients. They'll start to extrude protons, and very quickly, within a few hours, they can lower that pH from around 7 to 4. You can see that with other plants like corn and soybean. And a lot of this is mediated by a proton-pumping ATPase at the cell membrane. Okay, so plants are at the mercy of their soil, but they can change the pH a little bit. Also, cation exchange is just worth briefly mentioning on this lecture of biogeochemical cycling. This is another aspect of soil which affects its fertility. And what this relates to is the fact that soil particles, both organic and inorganic, have predominantly negative charges on their surface. And therefore, cations, or positively charged elements, have a tendency to stick to those particles, whereas negatively charged things don't. Okay? And the reason I'm mentioning that is then you often get a reserve of cations on soils because they're attracted to it and they'll stick to it, versus anions with a negative charge. And some of our most important nutrients are anions, like nitrate. They're repelled by soil, and therefore they leach out really fast, and that contributes to things like nutrient loading of rivers. So if it's a negatively charged anion, think about it's going to leach out quick. If it's a cation, it often will stick around in the soil environment. All right, so those are the first two parts of what I wanted to cover. I think that's all right. I can finish these other two parts that I wanted to talk about. Now we'll get into, now that we've had that brief introduction into what nutrients are important to plants and how they get into plants, wanted to mention the terrestrial nitrogen cycle, define what that is, and then talk about some of the anthropogenic changes that have occurred as a result of agriculture. There's been some really big changes as a result of agriculture. So nitrogen. Nitrogen, why is that needed at such high concentrations? Well, I already showed you one picture of it. It's needed for things like amino acids and building proteins. In plants, it's the key constituent of not only proteins, nucleic acids, chlorophyll, also a lot of the hormones that plants produce to regulate their life processes. They need nitrogen for that. And this is why nitrogen is so important for plants. Nitrogen is an interesting element, as we all know. Don't need to say this to you, but Earth's atmosphere is 80% nitrogen, but it's inaccessible to most of our life forms. You know, there's only certain bacteria that can grab hold of that atmospheric nitrogen and fix it. And these have been mentioned before in previous lectures here. Only bacteria and cyanobacteria, certain types of bacteria and cyanobacteria can fix and utilize gaseous nitrogen directly. And so there's several natural processes that fix nitrogen in the terrestrial environment. And people have tried to quantify how much nitrogen gets fixed by natural processes. And these estimates vary a bit, but I've pulled a lot of this from some plant physiology textbooks. It's generally said that natural processes fix about 190 trillion grams of nitrogen, or teragrams of nitrogen each year. And how does that occur? Well, about 8% of that occurs by lightning, which will cause things like nitric acid to precipitate and fall down to Earth. So about 8% occurs that way. You also have photochemical reactions, which are relatively minor. They account for about 2% of nitrogen fixation. And then finally, you have biological nitrogen fixation, which is being done by these certain types of bacteria. And this is the major way that nitrogen gets into the terrestrial cycle. This accounts for 90% of nitrogen fixation. It occurs when bacteria take atmospheric nitrogen, fix it into ammonia. All right, and just because this is such a major thing, this biological nitrogen fixation for the terrestrial nitrogen cycle, I just thought it's worth mentioning what are some of these bacteria that do this. Like I said, it accounts for most of the conversion of atmospheric nitrogen into ammonium. Most of the bacteria that do this process don't form symbioses with plants. They just live in soil, and they happily do this nitrogen fixation as part of their life's processes. So most don't associate with plants, but some do. And what I've listed here are just some in this table here. We're talking about symbiotic nitrogen fixation. So here we're talking about bacteria that do associate with plants. And we have some host plants here that do this symbiosis, and probably the ones that are most well-known would be the legumes. So a lot of legumes like clovers and soybeans, peanuts, just normal beans. These are all legumes that will perform or enter into a symbiosis bacteria. And by doing that, they gain nitrogen. Okay? And then these are some of the bacteria that perform nitrogen, or perform the symbiosis. And some of the most common ones are called rhizobium or cynorhizobium. Okay, and so what happens in this symbiosis is the plants develop a nodule, like I showed you on that first slide. The bacteria enter into that nodule. They begin to fix atmospheric nitrogen into ammonia. That passes to the plant. So the plant, therefore, has a major source of nitrogen. And in return, the plant diverts about 20 to 30% of its photosynthate to feed that bacteria in the nodule. So it's a very nice symbiosis that works for the plants that can do that. Take-home message, though, is that there aren't that many plants that can do this nitrogen fixation. None of our cereals can do this. It's just some legumes and some other plants like sugarcane that can do this. But it has been very important for agriculture, and prior to us putting down tons of fertilizer, it was the predominant way to get nitrogen into soils, and that's why I put the slide here. The cereal legume intercrop system still is one of the most important intercropping systems that you have. And so you can see a picture of that here. It's just you intercrop plants that can't fix nitrogen, like corn, with plants that can, like some type of legume. And so what that does is the plants that can fix nitrogen, they'll develop these nodules. They'll fix a substantial amount of nitrogen. That crop residue then enters into the soil in the next cycle and releases that nitrogen. And that can be then used up by plants that don't fix nitrogen, like corn. So this is a very important intercropping system. So here it gets us to what the full terrestrial nitrogen cycle is. And hopefully this will make sense based on that introduction to the terrestrial nitrogen cycle. Hopefully I can explain this rapidly based on what I've just said. So basically at the start of this, you have all your atmospheric nitrogen, and as we know, there's tons of that in our atmosphere. Some of that's going to come down through lightning and be able to be used directly by plants, but most of our nitrogen is going to get fixed by bacteria. And so that can be our nitrogen-fixing bacteria that are living in nodules of plant roots. So that accounts for some of this. But we're going to have a lot of nitrogen just being fixed by free-living soil bacteria. So what are they going to do? They're going to convert that atmospheric nitrogen to ammonia. Plants can take up ammonia, but it's very toxic to them. They prefer to take up nitrate. They can absorb ammonia, and if they do that, to avoid it being toxic to them, they'll put it straight into amino acids. But they'd prefer to take up nitrate. And usually when you add ammonia to soil, it very rapidly gets converted to nitrate. So plants don't normally have to deal with very much ammonia. It gets converted rapidly. So you have nitrifying bacteria that will turn this into nitrites, and then other types of nitrifying bacteria that will turn it into nitrate. And nitrate is, plants love to take up nitrate. That's their preferred form of nitrogen. And so then that will go up into these plants and be assimilated. So it's a key feature of this terrestrial nitrogen cycle. Then you would have some animals, like this little rabbit here, who might come along and eat some of that. Then afterwards, this plant dies. This animal is going to release some of this nitrogen as well, excrete some of it. It will be decomposed, all right? So it'll be mineralized, released as ammonia, and then it can enter back into this cycle. Also, sometimes when you have nitrates, it doesn't get assimilated. You can have denitrifying bacteria, which can release it back to atmospheric nitrogen. Okay? So hopefully that makes sense. Hopefully I didn't go through too quickly on that. But those are the chief features of our terrestrial nitrogen cycle. Now I just want to tell you a little bit about how we've changed that nitrogen cycle. Probably the biggest thing that we've, yes?
Sorry, just it's curious compared to the oceanic nitrogen cycle that say the ammonia is toxic where the take up nitrate.
Yeah.
The opposite is the oceans.
Yeah, I heard you say that on Monday.
Might be the solution here, but I thought that because it was closer to the ammonia is closer to the form that they make proteins out of.
Yeah. Yeah, I don't know if it's completely the opposite, but it's just often said that it's toxic to these plants. It is close, like they can put it directly into amino acids, the ammonia. But I've read and heard that, yeah, nitrate is the one they prefer, which would be a very interesting difference between terrestrial and aquatic. But we can talk about that. It could be, yeah. So anthropogenic changes, this is what I'm getting to now with changes that humans have made to this terrestrial nitrogen cycle. Probably the most significant one is the development of the Haber-Bosch process. Is this some process that you've heard about before? Yes. All right, so I won't spend much time on it. But this is a process that was invented in Germany in the early 1900s, in 1909 by Fritz Haber, and then scaled up to industry scale in 1913 by Carl Busch. So that's how this name comes about. And in fact, this wasn't done to create nitrogen fertilizer. This was actually done to create munitions, to create ammonia, a good source of it. And I'm not going to explain the process in detail. It's very complicated. This is an apparatus that carries out the Haber-Bosch process, but basically under high temperatures, about 200 degrees Celsius, high pressure, about 200 atmospheres, and in the presence of a catalyst, a metal catalyst like iron, you can take atmospheric nitrogen and convert it to ammonia. And before this, no one had done this. So when the Haber-Bosch process came along, this was a great way to suddenly have a source of ammonia. And you didn't have to rely on biological fixation anymore. You could do it industry scale. And so that was used for munitions, like I said at the start, but very quickly people realized, well, this is fantastic. We can use this for fertilizer. And it's credited, this process is credited with a lot of the green revolution that occurred starting about the 1940s and 50s when we greatly increased the productivity of our cereal crops, and the human population boomed from about 3 billion to 7 billion that it is today. To create those crops, to feed those crops and give them nitrogen, a lot of it came from this Haber-Bosch process. Worldwide industrial production of nitrogen fertilizer, now, so worldwide amounts of ammonia that we're producing through this Haber-Bosch process now amounts to about 100 trillion grams of nitrogen. And I just put, comparing that to the natural processes, that's pretty significant. Natural processes fix about 190 teragrams. This process alone is doing 100 teragrams. And we are growing more and more cereal crops as our population continues to boom. We'll probably have around 9.3 billion people by 2050 by conservative estimates. So we're going to need more and more fertilizer. And conservative estimates are we'll be producing about 165 teragrams of industrial produced nitrogen fertilizer by 2050. So at some stage, just that process alone will outcompete biological nitrogen fixation. And I just want to mention, not in much detail, but producing this fertilizer, doing the Haber-Bosch process, takes up a lot of energy. There's been some models that say it takes up 2% of the world's energy just to create all of this ammonia for fertilizer. And just burning of fossil fuels to carry out that reaction releases nitrous oxides, which are greenhouse gases, also releases some ammonia, so more nitrogen ends up coming down onto the land. So there's implications for greenhouse gases, but I won't go much into greenhouse gases because I know Robin's going to do that. And so I just brought up this image here. There's a lot of papers that I've looked at how much we have perturbed the nitrogen cycle in the terrestrial environment, and it's very startling, I think. Here's a paper from 2003 from Galloway, which just calculated nitrogen deposition per year, and you can see in 1860 versus 1993, and there's been massive changes, and a lot of this really accelerated with the discovery of the Haber-Bosch process. So you can see that in 1860, most of the world was towards the lower end of this scale, whereas now in 1993, and you can imagine now in 2015, this will be even more severe. There's all these regions of the world which are bright red, meaning they're up around the 2,000, approaching the 5,000 on that scale of milligrams nitrogen per meter squared per year. So there's been a huge change in the amount of nitrogen getting deposited. And this has consequences for nutrient loading. So this ties in with the lecture that we just had from Matthew. People try to estimate how much, so we're applying all this nitrogen fertilizer to our crops. It's misleading to think that those crops are absorbing all that nitrogen. Actually, the current crops that we grow around the world, like wheat and rice and maize, are extremely poor at taking up nitrogen. So in order for them to grow efficiently, you have to hyperload the soil. So they put tons of nitrogen fertilizer down, and a lot of that leaches out. And it's estimated that US dollars 500 million worth of nitrogen fertilizer just goes down the Mississippi River each year in the States. So there's a huge amount of nitrogen leaving these fields, making their ways into aquatic systems. And that's going to increase as we make more and more nitrogen and apply it. So that has economic consequences for these farmers who are wasting their money. But it also has huge biological consequences, as you know. Excess nitrogen in marine systems, in coastal systems, causes these hypoxic dead zones to occur. It often does. And we didn't have many of these in the past. We now have lots of these dead zones. They're increasing. The number of them is doubling roughly every decade as we apply more and more nitrogen in the form of fertilizers. And just briefly, how do these dead zones form? What happens is they usually form in the spring and the summer. In the sea, right where rivers empty into the sea, you have fresh water which is coming in into the sea right there. And basically during the spring, that fresh water forms a layer on the top and it cuts off the saltier water below. Okay, so you have that cutoff of the saltier water below. Then you have all these nutrients and all this excessive nitrogen that we're applying feeds. Is a lot of nutrient here. It feeds phytoplankton and they start to bloom. So you get these massive blooms that occur on the surface waters. The lower waters are cut off. The blooms occur for a while. They burn up all the nutrient, though, after some time, and then those phytoplankton die. They fall down to the base, and they get decomposed, and that creates this hypoxic zone to occur. As those phytoplankton are decaying, you get hypoxic zones where there's essentially no oxygen there, and that's the dead zone. And then once you have the dead zone occurring, then a lot of marine organisms die, like fish, and so you have a lot of fish floating up to the surface. So that explains what a dead zone is. Here's an aerial photograph of one of the world's largest dead zones. This one develops each spring and summer in the Gulf of Mexico, where the Mississippi River, which has all that nitrogen in it nowadays, dumps its runoff into this Gulf of Mexico. And here's a picture of it, of the dead zone. These are the phytoplankton that are growing and soon are going to cause the dead zone. In 2002, this Gulf's dead zone swelled to 22,000 square kilometers, and its size varies from year to year depending on how warm that year was. But it's a huge factor, this dead zone, and you get these wash-ups of dead organisms like shrimp and fish as a result. So there's a big ecological imbalance which is occurring here because of all the nitrogen that we apply. And by no means is the Gulf of Mexico the only dead zone. There's at least 200. Sometimes I've read that it's difficult to get the full number. So I think there's more than 300 these days. And they occur all around the world, and they're very much associated where humans live and where we're doing agriculture, because we're applying all of this nitrogen. And then there's just an interesting fact here, again, that relates to greenhouse gases, to nitrous oxide. There's been some studies that have been done of bacteria that live in these hypoxic dead zones. And it's found that microbial denitrification rates are about 10,000 times higher in these hypoxic zones. So they release a lot more nitrous oxide. So it could be that these dead zones are areas where we'll see a lot of nitrous oxide emission, which could contribute, to, of course, to greenhouse gas warming, greenhouse gas emissions. All right. Does anyone have any questions about that nitrogen cycle before I move on? Yes?
Sorry that I was just curious why India seemed to be enormous.
Yeah, how it had a higher rate. Yeah, I saw that. I think it's because there's always been a large population there, and they were applying a lot of manure and other ways of trying to get nitrogen into there. But I think it was just the dense population mostly that accounts for that. I think they do. I think they do, actually, yeah. So it's probably as a result of that. Yeah. Pete. It's a concentration thing, then, that you're saying. Ah, okay. That's good. That's good to know. Thank you. All right. In these last 10 or so minutes, I'm just going to spend a bit of time on the terrestrial phosphorus cycle. That was the last thing I wanted to cover today. And show you some anthropogenic changes also that have resulted from agriculture. Okay, and I think I can go a little bit faster through this cycle because it's a little less complex because you don't have nitrogen, you don't have phosphorus fixing organisms. Okay, phosphorus, why is this important? Incredibly important. Phosphorus is found largely as phosphate esters in all organisms. In plants, these phosphate esters play key roles in photosynthesis and metabolism. But also phosphate esters make up the sugar phosphate backbone of DNA and RNA, so you need that for DNA and RNA, and also phospholipids, which make up our cell membranes. As I say here, the phosphorus cycle differs from that of other major biogeochemical cycles, like the nitrogen one that we just went through, in that it does not include a gas phase. Okay, so this is strictly a sedimentary cycle. Basically, you have phosphates, which are present in rocks, which are leached out from those rocks via weathering, via rains, and then those phosphates, usually inorganic phosphates, well, inorganic phosphates always, distributed then throughout soils and water. And then plants absorb that inorganic phosphate from the soil. So you can see that it's a much different cycle than a nitrogen because you don't have little microbes that are fixing this element. And then unlike the nitrogen cycle that I just went through, the phosphorus cycle includes significant inorganic reactions. So a lot of the phosphorus that does weather out of rocks or that we apply by fertilizer, which I'll get to in a minute, undergoes mineral reactions and can become very unavailable to organisms. So a lot of phosphorus that ends up in soils is not available to organisms. It's very difficult for them to absorb it because of these mineral reactions that create insoluble complexes. And so here is a typical terrestrial phosphorus cycle. And again, there's no atmospheric component to this, so it's pretty simple to understand. We simply have our phosphorus here, which is present in rocks, which weathers out through rain. And so that can weather out directly into riverines, into rivers, out into the oceans, or it can end up in the soil. And that inorganic phosphate is available to plants. They will take that up into their tissues, and then when they decay, microbes will release phosphatases, which mineralize that, turn it back to inorganic phosphate, and that cycle can continue. Okay, but you also have a significant amount of phosphorus which is unavailable in soil, and that's because of these mineral reactions where phosphorus has formed insoluble complexes with things like calcium, and it's unavailable. Okay, so in a nutshell, how do plants acquire phosphorus from the soil? Well, they need it to be inorganic, first of all. They can't take up organic phosphorus. And that can pose a problem for plants because a lot of the phosphorus which is in soils is organic. In agricultural soils, 30 to 70% of all phosphorus is present in the organic form. In nutrient-poor grasslands, peat soils, and forest soils, this can be really high. This can be 80 to 95% of phosphorus is organic. So plants can't access that. They need to wait for that to be broken down by phosphatases that microbes produce to make inorganic phosphate. And so they're very dependent on microbes to do that. Some plants can actually release their own phosphatases, and I'll mention an Australian group of plants that does that in a minute, but the vast majority don't. So they're just relying on microbes to break down that organic phosphorus into inorganic so that they can use it. And rather than wait around, a lot of plants decide to invite microbes into their tissues themselves, form a symbiosis, and allow those microbes to break down, and actually fungi in this case, break down the phosphorus from organic to inorganic. And this relates to arbuscular mycorrhizas. Is this a word people have heard before? Or two words? Arbuscular mycorrhizas are simply a type of fungus which is very widespread in terrestrial environments. And these arbuscular mycorrhizae, they approach plant roots. Here they are on the outside of a plant root, and they will grow inside of it. So they will actually penetrate in. They'll penetrate within cells of the cortex, which is one of the cell layers on the outside of the root, and they'll establish a long-lived structure in there where they are basically, they will be breaking down phosphorus into inorganic form, bringing it in and releasing it to the plant. And in return, the plant is going to divert a substantial amount of its photosynthate to feed that fungus. So again, it's a symbiosis. In the nitrogen cycle, it was dependent on bacteria. Here, it's dependent on these arbuscular mycorrhizae. And most plants do this, so that shows you how important this is. 82% of all angiosperms, or the seed-bearing plants, the dominant plants, make associations with arbuscular mycorrhizae. And a large fraction of the fungal tissue is contained within these root cortical cells where you have this complex mycorrhizal interaction occurring. And it really increases the ability of plants to absorb phosphorus, this symbiosis. I told you I was going to mention a group of Australian plants that are really interesting for absorbing phosphorus. And this group of plants does it even without the mycorrhizal associations. The reason that we think these particular plants, which I'll show you on the next slide, have evolved their own mechanism is because Australian soils are extremely depleted in phosphorus. Australian soils